Does scientific genius suddenly appear in a sterile laboratory, or does it start on a forest floor with a simple, quiet sense of wonder? For Richard Ebright, it started here, north of Reading, Pennsylvania. As an only child, he didn't have much of a crowd to play with. Without enough kids to form a baseball or football team, Ebright directed his energy into the one thing he could do completely on his own. He became a collector. He gathered rocks, fossils, and coins with absolute determination. His mother recognized this drive immediately. She actively fed his intellect, buying him telescopes, microscopes, and mounting equipment to turn his solitary hobby into an exploration. Take a look at this list. By the time Ebright was in the second grade, he had tracked down and collected all 25 distinct species of butterflies native to his hometown. Raw curiosity is common in kids. But when that curiosity is actively resourced and encouraged by a parent, it builds the necessary baseline for a scientific mind. Eventually, Ebright ran out of local butterflies to collect. To keep him engaged, his mother handed him a children's book called The Travels of Monarch X. Readers were invited to help a researcher at the University of Toronto study butterfly migrations by attaching light adhesive tags to their wings. Chasing butterflies one by one was too slow, so he turned his basement into a mass rearing facility. He'd capture a female monarch, take her eggs, and raise thousands through their life cycle: egg, caterpillar, pupa, adult, just to tag them. But observation without feedback is tedious. Of the thousands tagged, only 2 were recaptured, neither traveling more than 75 miles. This graph shows why the local tagging method was flawed. The entire butterfly collecting season around Reading lasts for exactly 6 short weeks in late summer, giving him very little time to gather meaningful data. Mass data collection is an important step, but simply gathering numbers leads to a dead end if you lack a specific question to answer. In the 7th grade, Ebright finally put his work against his peers at a county science fair. His entry was a set of beautifully prepared slides showing frog tissues under a microscope. He lost. While everyone else around him took home a prize, Ebright sat there empty-handed. Ebright realized his mistake. Success at the fair required active experiments, while he had presented a static arrangement of slides. A display passively shows an existing fact. Science requires actively testing a hypothesis to uncover a new truth. That sting of defeat forced him to pivot entirely. He left passive collection behind to become an active experimentalist. For his 8th grade project, he tackled a problem. A viral disease wipes out nearly all monarch caterpillars. Ebright hypothesized that a specific beetle was carrying the virus. He tried raising caterpillars in the presence of these beetles. He didn't cure the disease or get definitive results, but proving he set up a controlled test to isolate variables was enough to win a prize. The next year, he set out to test the biological theory of mimicry among butterflies. This is a monarch butterfly. Biologists theorize that birds generally avoid eating them because monarchs don't taste good. Now look at the viceroy butterfly. Viceroys do taste good to birds. The theory suggests that the viceroy evolved to copy the monarch's visual pattern to avoid becoming a bird's dinner. Ebright designed a test to see if birds would actually eat monarchs. He found that a common starling bird would reject ordinary bird food and devour every monarch it could get its beak on, proving a crucial piece of the behavioral puzzle. Identifying a specific problem and designing a controlled test to answer it is the hallmark of science. By his sophomore year of high school, Ebright focused on a seemingly simple question. What is the actual purpose of the 12 tiny gold spots on a monarch pupa? Most people assumed the spots were ornamental, but Ebright and a classmate built a device proving that those spots were actively producing a hormone absolutely essential for the butterfly's full development. This specific discovery won him first place at the International Science Fair and granted him access to highly advanced professional military laboratories, like the Walter Reed Army Institute of Research. Fast forward to his time as a freshman at Harvard University. Using sophisticated equipment, Ebright began analyzing complex X-ray photos of this exact insect hormone's chemical structure. While looking at those photos, he made the ultimate breakthrough. Combined with his earlier findings, the chemical structure gave him the answer to a massive biological puzzle: how a cell can actually read the blueprint of its DNA. DNA is the substance in the nucleus of a cell that controls heredity, determining its form and function. By investigating a tiny gold spot, eBright showed how biological life reads its own instructions. eBright's path began with an insect collection and ended in laboratories mapping the chemistry of heredity. So, what exactly are the specific ingredients required to build a scientist? According to those who watched him grow, it takes two foundational elements: a first-rate mind combined with a deep, unyielding curiosity. The final ingredient is a specific type of competitiveness, the drive to do the best possible work, regardless of the prize. That is the formula. Find something you want to understand, start asking specific questions, and apply this exact blueprint to your own investigations.